This meeting is being recorded.
Hello, everybody. Good evening. So today we have very interesting talk by Hervé Isambert from the Curie Institute, and he will tell us about the causal discovery from multivariate information in biological and biomedical data. Thank you very much that you agreed to make a talk, and please start.
Okay, thank you, Alexander, for the nice introduction, and thank you all for being here. So I'll be telling you about our work in my group at Institut Curie. And so indeed, this is about causal discovery using multivariate information, and we apply it to both biological and biomedical data, as you will see. So as a word of introduction, I'm sure you all have an idea about what network reconstruction is. So basically, the idea is to look at some data in the form of a table like this, where the columns corresponds to some variables of interest, let's say genes, whose expression have been measured in different conditions or individual cells, for instance. So each row here corresponds to individual cells. And so, as you can see here, the data looks kind of a continuous data, but in principle, it can be any type of data. It can be categorical, even high dimensional, why not? And on top of that, you may also have some missing data, so some empty cells in this table. When you pick any pair of variables, so column in this table, you'll typically find some kind of correlations to some extent, given enough data. But obviously, most of these correlations do not correspond to direct interactions between those variables, but rather through indirect path, through some kind of graph. So the whole purpose here is to reconstruct such a graph, which basically disentangle this direct from indirect effect through a path. So as you can see, so we need algorithms, obviously, to do that. And there are actually a number of methods that try to build networks, similar networks. But most of this method, as we will see in a second, basically have a strong hypothesis, and so are basically restricted to a certain class of graphs. For instance, either a fully directed graph like this one, some Bayesian networks, or on the contrary, undirected graph where all these edges have no orientations, so-called a Markov network. And obviously, if you have to make these strong assumptions beforehand, you're not really learning causal relations, you're just assuming them. But indeed, there are methods that can actually learn causal relations from this correlation in such data. So in just one slide, let me briefly remind you or introduce you to these different type of approaches. So the first type of approach is this search and score methods that are based on trying to maximize some score, typically the likelihood. And they are quite nice, although searching the space of network can be very expensive because they are super exponential in number of nodes and edges. So it's really limited. This kind of exhaustive search is limited to very small networks, in fact. But there are interesting heuristics that can be used to learn even larger network. Yet there are typically strong constraints such that you're indeed building a network for which you assume that all connections are directed, for instance. That's this first class of method. The second class of method that builds undirected network this time is based on the inversion of the variance-covariance matrix sigma. which is well known to be related to the Laplacian of the graph, which basically give you connections between all directly connected variables. So if we assume that the top graph is actually the correct graph here, you see that trying to learn this directed graph through these second methods will actually learn an extra edge in the middle that does not actually exist, which basically closes this what we call the V structure. So a collider where you have two arrowheads that points towards a node. This is the signature of causality in these graphs, as we will discuss later. And the second method will actually close all these open V structures in the graph, in addition to removing all the orientations, because this second method indeed only basically assume that there is no causal relations. And then there is a third type of methods that will most be interesting for us, which concern these constraint-based methods that are based on trying to learn first some conditional independencies between pairs of variables. They are really nice methods because they can learn, in principle, a much broader class of graph that can have, as we can see here, both undirected and directed edges, as well as bidirected edges, as we will discuss in a second. So on paper, they are really nice methods. They have been, invented already some time ago in the late 80s, early 90s. But in practice, they have some problems too. So they're usually quite slow, but most of all, they are not robust to sampling noise. So although they have been proven to be correct in the infinite size limit, so when you have some oracle that tells you, correctly the conditional independencies in this data, but these are typically not robust to a finite size of the data sets, which is actually a problem. So we've worked on trying to improve the robustness of these methods by basically combining them with search and score methods, or I should say score methods. So to this end, we developed a MIC, which, as you can see, can be applied to different kind of data. So we've focused most on biological and biomedical data, but in principle, it could be applied to other type of data as well. And so here on this first example on the left, you see that we can correctly learn that most edges in these regulatory networks are directed. So these genes are essentially transcription factors that control the expression of another gene, which itself is often a transcription factor in this case. And while on the right-hand side, you see a more complex network, or a piece of a network here, where you can see undirected, directed, as well as these dotted edges that are actually bidirected. So we don't see the other end, but we'll see examples of these edges here. I should add that in all my graphs, the red color would be for activation or positive correlations, depending on whether they are directed or not. And blue are for repression or anti-correlation. So the way we learn these graphs and the fact that they have or not orientations is by assessing the probabilities to have an arrowhead or tail at either side of these edges. And if you have yourself some data like this, you can try yourself on our website here, mic.curie.fr. So here is a first example before we enter into the method itself. So here is a recent graph that we've learned from a fairly large data set. So here we are looking at nationwide healthcare data about breast cancer, about 400,000 breast cancer patients from the SEER database, which is one of the largest cancer database from the USA. So here, as you can see, the network is fairly dense and not completely obvious to read, maybe. But here we basically wanted to use this kind of data as a real world benchmark data sets. We've done a lot of synthetic benchmark as well, as I will show you later. But here, we really wanted to test on trying to use the largest possible or fairly large data sets, whether our method was indeed able to learn reliably causal relations in this kind of data. And clinical data is a nice real-world kind of data because you have a fairly good intuition about whether your prediction is likely correct or not. And indeed, as we will see in a second on a few examples, we could convince ourselves that in fact most of the predictions in this network were actually correct concerning the causal relations, I should emphasize. So 90% were clearly correct, and the rest is most of it also likely correct, as you will see, but sometimes kind of counterintuitive. So let me start with a counterintuitive example. But before that, I'd like to mention something a bit more technical. So the fact that we learn the orientation at either end of these edges means that quite frequently, in fact, we can be fairly certain that we have an arrowhead on one end of this edge, but we have no information basically on the other end. So this we call putative cause, because really we're not quite sure whether this is a genuine cause, which corresponds to a true tail and a true head. So this is what we define as a genuine cause, but the putative cause can either be indeed genuine or maybe due to some latent common cause, which we don't observe, which would be, somewhere in the middle of this edge. So it means that the other end can either be a tail or a head. So that's what we call putative cause, and this is actually quite frequent. So here we analyzing this data, these different causal edges, and I'd like to start with a kind of counterintuitive edge before showing you some more obvious causal edges. So this counterintuitive edge here, genuine cause here, concerns the relations between surgery and histology. Indeed, that's, as you may imagine, this is fairly counterintuitive, because histology is basically the type of cells that are affected by cancer here, while surgery is this act of the surgeon to remove the tumor. So obviously, we don't really believe that surgery is causing the type of cancer, which obviously started before the surgery. But nonetheless, we think that this is somehow what the data contains, because that's basically the way the histological type of these cells were collected. So it's mostly by surgery, or at least refined after surgery. And this you can convince yourself by looking at these different cell types. So indeed, our method, might indeed predict sort of absurd causal relations, as in this example, but that's, we believe, what's nonetheless in the data in this example. But as I alluded to initially, most of these causal relations that we infer are actually not counterintuitive at all. They are really expected, like metastasis indeed is the primary cause of death in cancer, which is well known. Some specific cancer subtype are on the contrary reducing the risk of death by cancer. This is known, and many other variables actually. Most of these relations are really well known and expected in this data. So this gave us some confidence that the method indeed is not just randomly guessing causal relation, but really managing to learn something from this data. There are also interesting missing relations or missing edges in this graph. So as we said, these graphs are fairly dense, but there are obviously many missing edges. So these missing edges, they've been removed, as we will discuss later, because we can explain the relations between them through indirect path. So in particular here, we saw that there is no edge relating insurance or the type of insurance of the patients and the survival. So we have 4 variables here that are related to survival of the patient in green here. And none is connected to insurance, although when we look at the correlations between these features, they are really biased. So having a good insurance really improves your chance of survival. But obviously, this is not the insurance itself that improves your survival, but the fact that you can get a good treatment if you have good insurance and can afford this treatment. And so this we can actually recover from this indirect contribution analysis.
May I ask a question? So,
sure.
So what is the input data for this analysis? Do I understand correctly that it's a kind of huge table? So it's a table data with something like 400,000 rows, and each node on the graph, it's a name of the column. Is it
exactly
correct? Okay, so we kind of have a table data, and you are managing to construct some graph first, and second, you show what are the causal relations. So,
exactly.
Okay. And this can be used for any kind of table data. Is it correct? Or...
Yes.
Okay.
Yes. To a large extent, yes. In principle, you can analyze any table data this way. Obviously, there are some constraints. I mean, it cannot be a huge table. So basically, the algorithm scales linearly in number of rows, in number of samples, and quadratically in number of nodes. So although there are tricks to improve this quadratic, because we're basically looking at all pairs of variables, but there are tricks for certain networks. We can improve this limitation. Yeah.
And may I ask another thing? So if we compute the correlation between the 2 columns, and can we think that something, if a correlation is low, then we do not have an edge?
Yes. I mean, if it's close to 0, you don't have an edge. But frequently it's not close to 0, and nonetheless you don't need an edge. That's the.
Okay, I see. Okay, thank you very much.
Thank you. So don't hesitate to interrupt me, and I'll be happy to answer your questions.
Thank you.
Okay, so that's the example I wanted to start with. So probably for the interest of time, I'll skip the rest here. We also look at socioeconomic variables in this network. So there are quite interesting results, but I think I'll go more to the method now. And so the first question we may ask, concerning the methodological part is really whether one could hope to infer causality from just looking at correlation in this data, because as all of you know, indeed correlation typically does not imply causation. And that's actually true for 2-point correlations. One of my favorite example is between the barometer and the weather. Indeed, they are correlated. The barometer has been invented for that, and the barometer tells you the weather that will happen in a day or two. But obviously, it's not causing the weather. So even this time delay is not really, sufficient to explain that the barometer would cause the weather, which nobody believes, obviously. So indeed, in such an example, what happened is that these two variables, barometer and weather, they are both responding to a third variable that is not directly observed, that is the, whether, atmospheric depression, or that to which the barometer will respond faster than the actual weather that will take time to arrive. So that's basically the causal relations between these two variables, which is mediated by this hidden common cause with some different delays between the two. The surprise, though, is that when you go beyond 2 points, so if you go to 3 or more points correlation, those multiple point correlation might actually imply some causation, and so there is some signature at some level that in the correlation themselves that tells you that this cannot be explained without causations between those variables. So this sort of reasoning started already some time ago with Hans Reichenbach, a physicist and philosopher, I think, that proposed this common cause principle, which basically tells you that when two events E1 and E2 just that have no reason to co-occur indeed happen simultaneously, our intuition and daily reasoning really infer that there must be a common cause to explain this coincidence, basically. So, in fact, in our daily life, this is something that works very well, as you realize. But in practice, when you really look at data for which you have no prior knowledge or conception, it can be shown that this is not really operational. So when you have 3 variables, E1, C, and E2, that are consistent with the common cause relations, they are also consistent with some sort of a mediation diagram where E1 would actually cause the middle variable that now we call the mediator, and that itself causes E2. And so there is no way really you can distinguish between these different very simple diagrams unless you have some prior knowledge, which obviously we have in our daily life. We realize that some things are possible and some other things are not by our past experience. But when you don't have this prior knowledge on data, basically there are many things, this common cause principle is not really operational. What is really different is something that one could call the independent cause principle. It's not actually how it's called, but let's simplify it this way now. It's when you have 2 causes, C1 and C2, that actually somehow cooperate to explain one effect. And so the idea is when you have 3 variables, 2 of them are independent, C1 and C2, and yet they are both strongly dependent or correlated with a third variable E, then the only possibility is that E is not the cause of C1 or C2, because otherwise we would have some kind of correlations between C1 and C2. And if you can observe in the data and convince yourself from the data that this is not the case, then E has to, be at the at this collider position where two arrowheads are pointing to it. One strong limitation, though, is you can't really exclude by just looking at these three variables, C1, E, and C2, that there is yet other variables like L, a common cause between C1 and E. So if you don't have data on L, it's always a possibility. So you can't really be sure that C1 is causing E. What you can learn from this data is that E is not causing C1. So that's the conclusion of this graph. Okay, so the idea is we will try to learn from the data this kind of relations. And so obviously this V structure will be the signature of causality in this diagram. But as we just said, you can also have a bidirected edge, which are basically when you have two V structures that share an edge. So here we have two V structure, X, Z, UI, and Z, UI, W, that share this edge, as if there was some common cause L not observed in the data. So this constraint-based method that I mentioned earlier, they work all more or less the same way. They start from a complete graph, and in the first step, they will remove as many edges as possible that can be removed through conditional independence. So, for instance, here we can convince ourselves that X and UI, although they might be correlated, they become independent or conditionally independent when we condition on W. And so we can remove this edge by conditioning on W. And the same for all these dotted edges. So at the end of this first step, you have what we call the skeleton of this graph. This is when you have removed all the edges that you could by trying to find conditional independence. Then in the second step, you try to find this V structure signature. So this signature of causality in this data. And for instance, here we could learn these two V structures that actually share an edge. So we learn also this bidirected edge in the meantime. And finally, you can also what we call propagate some of these early orientations, because once we are convinced that we learn all the V structures, we can. And so it means that these edges that are downstream of V structures, if they are not V structures, we can infer with some hypothesis that these edges have to be oriented the other way. So that's how we propagate some of this orientation. So as I alluded to earlier, this method they are not recent, and they are on paper they are really nice, but in practice they are not really working that well. So, and the reason is that they are not really robust to finite size of the data set. So our contribution was to make them robust by combining those causal discovery methods with the information theory.
Okay, may I a little
Sure.
bit ask? So, is it possible to give some maybe more intuition on? So, for example, on each step. So first, conditional independence. So what are we doing? We erasing some edges.
Yeah.
Because of what's our kind of intuition to erase these edges?
Yeah. Okay. So the intuition is that we will erase all the edges that we don't need to explain this data. So we will try to. The idea is that some of these correlations between variables will be due not to direct connections between these variables, but through indirect effect through other variables. So when you condition on W here, it means that you can infer that the information between X and Y somehow is transmitted through an indirect path through W. So by conditioning on W, you intercept this information, and then you have now in conditional independence between X and Y, which tells you that you don't need an edge between these two nodes, because otherwise conditioning on W would not, be sufficient to make these two variables X and Y independent. Okay, so that's the idea.
So these conditional conditions, they can be calculated from this tabular data.
Yeah.
So it will be just from correlations, or you need something more, something like mutual information between these rows or something?
Right. So I mean, there are different ways. So the typical methods. So in this early paper, the idea is to look at to have a statistical test for conditional independence. So that's the basic idea. The problem with that, which explains why these methods are not robust, is that conditional tests basically slice your data. So imagine you start with a seemingly a large data sets of 1,000 sample, but if you have to condition on one variable W which has 10 levels, say, it means that you will basically slice your data in 10 smaller pieces, so on average 100 samples per subset, on which you'll try to pass this independence test. So now imagine that you need not one but several conditioning variable, let's say W and Z. Then if W and Z have both 10 levels, it means that you end up slicing your initial data sets in tiny subsets of on average 10 samples. So the problem with this method is that there is always a way to slice your data, so to pick some other variables, and because there is this combinatorics, there is a huge number of possibilities to combine the conditioning nodes so that you slice your data in such a way that X and Y appear independent. But this is just noise. I mean, this is just finding a way to slice this data so that on very small subset this variable appear independent. So that's the origin of the lack of robustness of these methods. And I should add that, in fact, this is very easy to find such a combination of variables. And so, many of these, so most of these, initially we start from this complete graph, so we need to remove, in fact, most of these edges to find some reasonable sparse network at the end. And so each edges that needs to be removed is prone to this error. And so fairly frequently you will remove, unfortunately, one of the few edges that you should actually keep. And after making this early mistake, you end up with some kind of snowball effect where you try to keep edges that you should remove to compensate the early error. So that's the origin of the problem with this, the lack of robustness of these methods. And in order to improve that, we coupled them with information theory, as I will discuss right
sorry,
now. Yeah?
Please, can you come back to the previous slide? So when you calculate this conditional, do I understand correctly that it's better for, I mean, theory and practice where our variables are discrete and they have not so many values? So, for example, in your example, if it would not be 10, but only 2,
Yeah.
This is correct, yes?
Yes, in principle, it's in principle, but eventually it will nonetheless become a problem.
Yeah,
as soon
sure,
as you-
sure. Yeah, just
but for the illustration, yes, it's clear that this is a problem with in particular discrete variables with many levels. So in fact, we have a way to deal with this particular problem as well, which is specific problems in this sort of analysis.
Okay, and so another question. So do I understand correctly? So this step, we can say that it's kind of tries to preserve only something like direct interactions, and all indirect are removed. This is
well, all indirect, we hope, will be deduced from the direct graph. So basically, we hope to be able to read this network so as to learn the direct effect as well as the indirect by just looking at the network, which is another actually serious problem because most of this method are not really reliable either on indirect effect, but that's the hope. The hope is to be able to read both the direct and indirect effect by looking at the final network. Yes.
Okay, and there is a question in the chat. So just a moment.
Yeah.
You use do operator introduced by Perl for causality model or not? There is some question.
No, so that's a good question, but do operators are basically to try to estimate the causal effect. So give a number on how much UI would vary when I vary X or vice versa. So that's really to quantify the effect. This is not what we're trying to do. What we're trying to do is to basically first learn the graph. Which node is actually causing which other nodes, and to what and so we're trying to learn this is what we call it causal discovery. We're not trying so far to quantify this effect, which can be done, for instance, with the do calculus, as mentioned in the question. Yes.
And another question. So the step number 2, orientation of these structures. So can you give some, again, intuition what's happening here? So how do you introduce this orientation? What is the intuition behind?
The intuition, as I mentioned earlier, is that this V structure is the simplest structure that carries this signature of causality. And it means that we really need to put these two arrowheads here when we want to model the fact that X and Y are independent, so truly independent. Just imagine, just imagine that Z is a car, or whether my car is broken or not, whether it runs or not. So obviously, the fact that my car is running or not, let's say not running, may have different causes. So let's say the battery is dead, or maybe the pump, the fuel pump is dead. One can hope that these two possible causes are independent, but obviously both can lead to my car not running. So that's the idea. When you have a strong connections between X and Z and UI and Z, but yet no correlations between X and UI. So this is the signature of causality, meaning that Z cannot be a cause of X or Y, because if Z was the cause of X or Y, then X and Y would be correlated to some extent. So we'll have information passing through Z.
That's the point. So I understand now. Yeah. So
Yeah.
very nice idea. Yeah. So
Yeah.
if it would be in other direction, X and Y should be correlated. And if we see,
Exactly.
yeah. So yeah.
So
Thank you.
basically, as we said earlier, we don't really know whether X is causing Z, but we know that Z is not causing X. So let's say if we do an experiment, if they are not cars but genes, if we do an experiment on knocking out Z, we expect no effect on X and Y. While if Z is really a cause of X or UI or both, then we expect to see something on X and UI when you knock out Z. So there is a prediction here that can be tested.
Yeah, very nice. Thank you. Yeah.
Okay, so that's the basis of all this. But as we just said, it's really nice on paper or on when you talk about it, but in practice it's not really working so great when you have a finite data for the reason we just explained. So we try to somehow make it work or make it work better on finite data, and for that we use indeed information and multivariate information more specifically. And the idea in one sentence is to use conditional mutual information to find conditional independence, but we don't want to run into the same problem as the traditional method that try to find directly the combinations of conditioning variable AI here that make this conditional mutual information very small. So because we want to avoid that, what we want to do instead is to basically collect significant contributors. So we want to avoid to have something fairly small here directly, because this is really prone to be confused with the noise, basically. But the idea is to find significant contributions. So this is above noise that we can somehow measure. And it comes in the form of this 3-point information term, which is kind of a generalization of 2-point mutual information. And so we will subtract from the 2-point information between X and Y that obviously contains all the direct and indirect effect between these two variables. We will subtract this 3-point, which basically corresponds to the contribution of A1 to this mutual information. And so once you subtract this first term, typically the residual here is still significant. And in that case, you need to find a second strong contributor, again above noise, and you can in principle iterate that. So we're not trying to find the combinatorics, we'll try to find the best next contributor, basically. So it's some sort of greedy approach where we don't know exactly where we go, but eventually if we find something small enough, we have some confidence that it's indeed not random, but due to the fact that we collected significant contributions. So that's the first idea, and this is how we basically prune this graph by looking at this more robust conditional independencies by collecting this indirect effect. Then in the second step, we use again.
Yes? So when you write this formula, you have this term AI, A1, A2. In our case, it will be again the rows, yes?
Yes.
Okay.
It's still the variables, yeah.
Okay. Thank you.
Exactly. And you express it, looking at probabilities over the data set, basically. So in the second step, we will again use the 3-point information. But as I did not mention earlier, this 3-point information, unlike the 2-point information, which is always positive, as you probably know, the 3-point or more points multivariate information can be positive or negative. And so when it's positive, it's a contribution. And when it's negative, well, it's not a contribution. And what it means is that indeed this third variable, if it's not a contribution, this value, when it's strongly negative, gives you some confidence about the fact that you just learned a V structure. So it means that Z is not a contributor in that case. Another way of saying it is that, if you pick here Z, if you subtract this negative value, it means that in fact, instead of reducing the mutual information between these two variables, you are actually increasing it. As if, for our car, once I know that the car is broken, so when Z is my car is not working anymore, basically there must be a reason, and the reason is, to be simple, either the battery or the pump or maybe something else. But so basically, and since when I found the battery, it's unlikely that the pump just happened to be broken at the same time. It means that I learned that my battery and my pump are somewhat uncorrelated or anti-correlated rather in this example. So I learned something about the state of my possible causes X and Y once I learn Z. So that's what happened if I put this term here. So obviously this is not what I want to do, but but I can use these negative terms to learn, with a score, this signature of causality. So this is something we showed almost 10 years ago now, that we can really use to learn, to get high confidence on the orientations of this graph. But as you realize, this is how you learn the head of these edges. If you want to learn the tail, you have to use the next step, which again use the positive 3-point information once you already know orientation. So if I know that X, Z, V has an arrow pointing towards Z from X to Z, and that the 3-point XVZ is positive, then it tells me that, here, on this side here, it cannot be a arrowhead, another arrowhead, because otherwise, I would have found it with a strongly negative mutual information. So if it's strongly positive, it means that here I just learned a tail. So that's how I learn the tail on my edges. And once I know on a given edge both the head and the tail, I can reasonably assume that I found some causal relations genuine causal relations between these variables. Okay, so let me show that indeed it works. So here are synthetic benchmark now. So we know the ground truth, and we can see how, using this information measures improve our predictions compared to the methods that don't use the same sort of information ideas.
I'm sorry, when you say we know the ground truth, so how it is achieved? What are the methods to create something where we know?
So here here are three classical benchmarks. So they are Bayesian graphs from which you can hide some of the variables. So you create these data sets with latent variables as well, but starting from a Bayesian graph. So some graphs are more easier to learn than others. So for instance, the first one here is kind of easy. There are 500 parameters, but you see that the second one on 1,000 and the last one has 100,000 parameters. So the more parameter, basically the more difficult these graphs are to learn. And you see that indeed for the simple graph, our methods reach f-score, reasonable f-score, close to 1, but obviously not equal 1, but around 0.9 or so fairly good result. But yet the classical method eventually catches up, but it needs significantly more data to do that. While, as you can see, for more difficult benchmark like the last one, we don't do perfectly, because here we only looked at 50,000 samples, so it's clearly not enough to learn all these parameters or all these graph. But the classical method has even more problems to do it. So definitely coupling this causal discovery with these information scores definitely helps.
Absolutely, and related question. So, is there something like a way to create a new kind of artificially? So something like I can't imagine how we can say that artificially create a graph and say this is a kind of directed.
I mean, all these benchmark are synthetic benchmark are done the same way. I mean, you assume a graph, so with some specific properties like degree distribution and strength and number of parameters, levels, and you name it, and then you're trying to learn it from an increasing amount of data, and then you compare the different methods.
Okay, thank you.
Okay, so that's all for the methods and benchmark. I'd like to show you some applications and probably go fast on the first one and then try to move on to temporal data if I have enough time. So here is one of the first example on single cell expression data that we look at. I just show this example because here our collaborator wanted to show the difference between the correlation network in gray, which, as you can see, connect most of these nodes. So it's correlations do not distinguish between direct and indirect effect, while our network or other method to learn the direct connections will show you something which is much sparser, but yet that can be interesting. And in our specific example here, we could actually learn this new hub marks for this particular problem. Another recent study that we did on single cell expression data is related to a cellular therapy, actually against multiple sclerosis. So in this mouse model, so these are mouse that in which you can trigger the disease, and after about 2 weeks, basically they are paralyzed. But in this, our collaborator, Simon Filatro's lab at Necker Institute in Paris, have discovered that you can actually cure these mice by injecting them some other cells that are called B-Rex, that just happen to that have been made to express this IL-10 gene in fairly abundant and stable conditions. And so by just injecting these cells in the brain of these mice, seems to cure them after a few days. So this is really impressive. So these mice that really looked in rather bad conditions are basically regaining all their normal life after just a few days of injecting these cells. So we were really fortunate to be able to look at the data. So the data was this single cell RNA-seq data. And Louise Dupuis in my team that analyzed this data first identified as these injected B cells, B-Rex, mostly affect the microglia, which are the other type of cells in the brain whose job is normally to protect the neurons and clean the brain, essentially. But in this disease, these microglia, switch phenotype, and instead of protecting the neurons, they actually attack them. So this is actually an autoimmune disease where the cells just fight against other cells. And so then we analyze how the treatment, so the injection of this BREG, somehow managed to reset the expression of this microglia from this autoimmune disease state to their normal protective cells. And we could first, we can predict some of main regulators in the graph, gene networks that the treatment basically downregulated, and then this effect obviously propagates in the rest of the network. I have another example, but probably I will skip it for the interest of time. This is to extend our methods to reconstruct very large network. So we can now, by distinguishing transcription factors from non-transcription factor genes, we can now reconstruct network fairly reliably with thousands of genes. But I'll skip this, and because I'd like to tell you a few words on the extension of this kind of analysis to time series. So this is also not a recent field of research. So it has been pioneered in particular by Granger, who proposed to basically test whether looking at the time traces of different variables, we could not deduce that X, for instance, might be causing UI by just looking at this signal with maybe some kind of delay. And in the first version, the idea was to try to find the best regression of the value of UI at time t, given its past and the past of this other variable X. So if you can convince yourself that you're better off predicting UI by including the past of X or not, you may conclude that X might actually be causing UI. To go beyond this linear assumption here, later in the early 2000s, Schreiber introduced this concept of transfer entropy, which actually is nothing else but conditional mutual information. And so because information can be, is very good actually at capturing signal, not only for linear, but also nonlinear relations. This is a fairly nice extension of Granger idea. And so the idea is to compare the transfer entropy from X to UI or UI to X. Obviously, as you realize, these formula are not symmetric. So any signal, causal or non-causal, is expected to have different transfer entropy. So really, although it's not always used properly, you can't just estimate the transfer entropy in either direction and then conclude on causality. So really the only way, and already in Schreiber paper, original Schreiber paper, it was rather clearly explained, but that then probably forgotten by many readers. The idea is that you can only conclude something by trying to find some vanishing transfer entropy in one direction while the other is positive. So this is something that tells you that, as we saw earlier, that UI may be caused by X, but probably not the other way. Yet obviously you're not quite sure that X is causing UI, because we can always have this latent variable in the middle, like this atmospheric depression in our early example, where X is the barometer. So X definitely has some signal earlier from UI, the weather. but in fact the barometer is not causing the weather, although this approach would tend to suggest that the barometer may cause the weather. Is just because there is some latent effect that causes both variable with a further delay. So my first question here is really what's the connections between this temporal causality that we just defined and the early graph-based causality that we discussed. As you realize, the transfer entropy idea is really nice because it's also based on conditional mutual information. So there ought to be something there to discuss what's the relations between these two quantities. It's not so clear that they are equivalent. In fact, they are not. But one happens to imply the other. And to show this, the idea is to reason on some kind of time unfolded graph. So now the previous graph, we have different points, different nodes for past times, t prime, of each variable. So ui at t and t minus 1, minus 2, and so on. Same for x. And so we can try to learn some graph, time unfolded graph, using our early approach. And then what we can conclude is that in order to have a vanishing transfer entropy or conditional mutual information here, it means that this dotted edge, so it's between x, this variable here, and the past value of ui, so the light blue. So in order to have something vanishing here, condition on these nodes, you ought to have no edges, no dotted or dashed edges here, because any of these edge would make this non-zero. So this means that there is no edge here. But it also means that here you need a V structure, because if this, so these edges, these arrowheads come from reasonable assumption from time. So we know that ui at time t cannot cause anything at time at earlier time. So all these edges here ought to have arrowheads, but this edge here could be any directions. And so we learned that this direction, this edge should have an arrowhead also here, because if it had no arrowhead, then this wouldn't be zero. Because we will have information transfer through UIT. So clearly we have these implications, but as this counterexample shows here, the converse is not true. So you can have, so for instance, this graph, we have clearly some causal relations that we can learn with the graph methods, but it does not imply vanishing transfer entropy. So basically we can't, so you can show theorem that shows that temporal entropy will imply this graph-based causality. Temporal causality will imply this graph-based causality, but not conversely. And so what it really means is that the temporal causality is somewhat too restrictive, and it may miss actual causal effects like this one here, that you can learn with this graph base. But the conclusion is nonetheless that there is no different causalities based on time or based on graph. We can assume that there is only one causal concept, and that's probably the relations with the early temporal causality introduced by Granger and Schreiber. And so we have applied this, and we'll finish with this last example. So we've applied, so we've adapted our methods to learn this graph based on temporal time, so I don't go into the details here, but the idea is to apply it to time series data, and we've applied it to essentially movies, so video images, and we've used it to analyze time-lapse images of live cells that here corresponds to tumor microenvironment that is reconstructed ex vivo. So in these small microfluidic chips, you can have you can put different cell types to reconstruct some tumor, artificial tumor, with cancer cells, but also immune cells, because we're looking at eventually at immunotherapy, where the immune cells will fight cancer, essentially. So we need immune cells, cancer cells, and we may also have other cells like CAFs in particular that are cancer-associated fibroblasts. And so we've analyzed this data from the Maria Carla Parini lab, and the idea is to look at many of these images. So we look at more or less 50,000 of those images, extract some features around cancer cells and immune cells. So first segment these images and then track the cells, and then from this data define many different features that can be of interest. And at the end of the day, we were able to reconstruct a graph that now looks like this. So now you can have several edges between variables, because there might be different effect at different timescales, and these numbers here are corresponding to the lags between the cause and the effect. So obviously, by analyzing this data, we recovered that the treatment indeed improve the apoptosis of cancer cells, which was the goal of the treatment. So that's not new, but obviously we recover it. But we see that treatment has also many other effects on different variables. What we learned as new is that the CAFs, so this third cell type, in fact, has a direct protective effect of the cancer cell. So until now, it was thought that the CAFs were somehow indirectly decreasing the effect of treatment, but our analysis suggests that in fact they have some more direct effect on protecting cancer cells independently from treatment. But this graph, as we said, they can have different edges, and as you can see here, they can have edges with different colors also, so they can have antagonistic effect at different timescales. And we have a number of examples like this, in particular here between division, so cell division, and eccentricity. So obviously, when the cell divide, it has to become eccentric. So with 0 time delay, the dividing cell becomes very eccentric. But what we could recover, which is actually known by the biologists, is that if you look at these cells that will eventually divide in 2 or 3 hours, they actually become very round. And this is when they replicate all their biological material. So basically, there is some decision, once the cell has made the decision to divide, which could be, some latent variable that we do not directly observe, of course. Once this decision is made, then, the cell becomes very round, and then 2 or 3 hours later eventually divides. So you see that this method can capture these different causal or non-causal effects, including delays. And with that, I think I'll conclude, because I'm way off my time. And I'd like to thank the students and postdocs that have worked with me on these different topics. And so this is the group as of a few years back now. So many of these students have left. I mean, so I showed, for instance, the work of Marcel, Vincent, Louise, and Nadir. Louise is still with us, actually. And so the group now is made of these people. We'll have two new PhD students at the autumn, and we're actually hiring. So feel free to contact me if you're interested, and I'll be happy to discuss with you. With that, I thank you again for your attention.
Thank you very much, Hervé. So great talk. Thank you very much. There are some questions in the chat. So let me, maybe you can keep the presentation if it's possible.
Okay, I can. I was just looking at the chat, but
okay.
Maybe you can pick and choose for me. Go ahead. Yeah.
I will read the question. So the first question, how this method deal with spurious correlations caused by compositional effects, for example, in microbiome data?
So spurious correlation. Okay. So it depends what we call the spurious correlation. So spurious correlation can be, any indirect effect. So this is actually the aim of these methods, to disentangle direct from indirect effects. So, but obviously you can also have biases in the data. So I haven't shown you examples, but this is very frequent. So typically, the first thing you find about data sets when you have a new data set are the biases. So some experimental conditions that was not set properly, or that somehow change over time, and so all this control that you can expect from this data will, if any bias obviously will show off in these networks. So in that case, you need to go back and maybe correct or pre-process your data to correct this effect, or maybe keep it as is, but showing that you have some significant bias in this data. So I could show you some examples, but probably we need to go to other questions.
Another question is, what is the ratio between number of samples and number of features to run this method effectively?
There is no absolute rule. So as I said, so the complexity is essentially linear or sublinear, depending on the type of data, in terms of sample. So as you saw, you can have quite a few samples, but the method is quadratic in terms of variables. So the basic method cannot scale beyond a few hundred nodes. But for certain data, like gene regulatory networks, we actually can use prior to somehow simplify and to not test all the pairs, because we know that some pairs of variables of genes, in that case, cannot directly regulate themselves, because they are not transcription factors or any kind of regulators. So, as using this assumption for specific applications, we can really scale the methods to much larger networks, and that's what we do for gene regulatory networks, where we can have thousands of genes, but most of them obviously are target genes. They are not regulators, but we can still have a few hundred regulators.
There are another question. So, colleagues, just for the moment, now I enable, you can unmute yourself and ask the question by voice. It's up to you. If you can, it's better you can write a question in the chat, or you can ask by voice. Up to you. Just what's convenient. So Another question, what algorithms for graph structure learning are the most powerful? Is it LinGAM or not at all?
So Lingam, I'm not. I should double check. So you'll check for me after. But I think Lingam is one of these algorithms that uses kind of strong assumptions about the structure of the underlying data generation mechanism. So providing you are looking at data that have been generated or that is reasonably modeled by this generation, it can work. But in for any other data, it may not work that well. So there are a number of techniques that use, for instance, the linear noise assumption, or this. So there are even methods that try to infer causal relations by just looking at 2-point correlations. But for this, you need additional assumptions. So as soon as these assumptions fail, these methods, they do not work so great. So we tend to favor methods that do not rely on these assumptions, because when you look at actual data, like biomedical data or biological data, little is known, and these strong assumptions about the structure of the noise, etc., are typically not met. So this is a strong limitation.
Thank you. Another question from Valery. Valery mentions that there are different type of causal relations, direct and indirect, contributory, necessary, sufficient, proximate, and so on. And his question is, your causal relations are of that kind or some other, and how it's related?
So I'm not sure.
It's some, yeah.
Yeah, so I'm not sure it directly relates. I'm not sure to what you allude to, but so I try to make the case that basically there are different type of relations, causal and non-causal relations, but often only partially. I mean, you can only learn partially this the nature of these edges. So you can learn, for instance, an arrowhead on one side of this edge, but you have you can't infer anything on the other side of this edge. So this is what we call the putative cause. So putative cause, it's maybe a cause, maybe not a cause. So it's the first step that from which you actually learn that something is causal, but you're not quite sure yet. So if you remember my talk, the first thing you learn is that something is not causal. So the V structures tells you that the tip of the V structure doesn't cause the other two. But in order to deduce that the other two variables may actually be real or genuine cause of the tip of the V structure, you need more. You need more than 3 variables. You need other variables. If you're interested by this, you can look at our paper, Ribeiro Dantas 2024.
Thank you. Another question appeared. Are there some metrics about to measure this graph looks like an interaction network, or this is not? For example, for single-cell RNA sequencing data regulatory network, can we say this is kind of this type, or this is not?
You mean in the topology of the graph or something like this?
Yes.
Yeah.
Anything nice as well.
So yes, I mean, to some extent, yes, but many biological networks will have this hub, for instance, so hubs are genes that will regulate many other genes, hundreds of other genes. So this is quite frequent in biology. While you're typically not regulated by hundreds of regulators, but just by a few tens at most. So there is some asymmetry between in degree and out degree, and you can look at this in terms of topological measures of this graph. So this has been done extensively, I must say. Yeah, I think now we are beyond this qualitative description of this graph. We're really trying to, learn the actual regulations between variables, let they be genes or other variables, like in biomedical data. It can be much more abstract than genes in cells.
Yeah, thank you. And another question, so from Andrei, if we study, is there a link from X to Y? Do other features important for this analysis? So Andrei, as for instance, so Hervé showed that there is a kind of V structure, yes? So it's one of the points that if you have three variables, then it's more easy to learn something. So Andrei is commenting.
Yes, please. But I think this question is quite simple and it's clear. I think yes, and so of course, for studying the relationship, the causal relationship between X and Y, other features are very important. Yes, so we cannot say that only X and Y columns are enough. They are not enough.
No, they're not. Yeah, exactly.
Okay. Yeah, are not. So okay, but and of course, what's maybe how to do if for example the X is in the past and the Y is in the future. For example, there are different features, but X in the past, Y in the future. But some algorithm, causal discovery algorithms, finds the link from Y to X. So from the future to the past, which is wrong. Of course, which is wrong. That's what to do. To find more data, to enlarge data set, to maybe find more features, what to do.
So what we ourselves do is that when we look at temporal data, like in the example you mentioned, we basically use these strong assumptions that we have that present cannot cause the past, or future cannot cause the past. So we assume that we have arrowheads that are pointing toward the future. But again, it does not exclude that you have another arrowheads pointing towards the past, which corresponds to a latent variables that explains the relations, but with different time delay, like this barometer weather example that I was mentioning early on. So it's well possible that you have a bidirected edge with a delay, so that points towards the future and towards the past. But the interpretation is that the common cause of the two is started yet before the effect. So it should be a bent edge, if you want. Yeah.
So to look for other features, other latent maybe features that were not processed. Yes, to add additional information. In other words, yes. And I have one more maybe question is regarding gene regulatory networks. We had a gene. You have a slide there where the graph is not directional, indirect graph. Yes, but gene regulatory network without a direction. Yes, just we are about only is there a relationship between, and what's the power of a relationship between maybe one gene and another gene. Yes, And so, as I know, now, one of the methods is to maybe to run a random forest algorithm, where we consider one gene as a target, and the other genes are predictors, and their feature importances are the values maybe of the row in this gene regulatory matrix. Of course, this is quite unstable because features are correlated. Of course, there are some different problems. But you mentioned some other methods, if I understand you correct. Some other methods because maybe that are more powerful than just to run random forest and you using its feature importances.
Yes. So this is Genie 3 that you're describing, I think.
Yes, Genie 3. I described Genie 3. But maybe you mentioned some others in your presentation, or not?
Yeah, so
maybe you talk about something else.
The method that we develop is not based on this.
But is it causal or not?
Tree forest. Sorry, what?
Is it causal method or not causal? Genie 3 is not causal.
Yeah, I think it's not causal.
About causality.
As you said, it's a random forest. It's not causal. Yeah.
Yes, just about relationship, about correlation, not causation.
Exactly. Yeah? So basically, the method that we use starts, as you saw, without, start by looking at conditional independence without looking at causal relation. So we first learn the skeleton, which has no orientation. And then in the second step, we'll try to infer, when we can, some causal signature on top of this skeleton. And for instance, we actually we try to trick our approach by looking at data, synthetic data that was not causal. So we built some spin models where these nodes are spins that interact with a number of neighbors. So you can build any random networks of spin like this. And then you generate as many configuration as you wish of this spin network. And there is only one thing you're sure is that there is no causal relation. There is only free energy in this or interacting energy between these spins. And so the idea is to see whether causal discovery methods can be fooled by this kind of data, which has this network structure but no causal relations, because we build it this way. And we could actually show that the method indeed realized that there is no causal relations in this kind of data. So it learns the skeleton. We applied it also on protein structures. When you look at connections between residues in 3D space. And for this, most of the connections have no causal relations. Actually, we have example, there is absolutely no causal relations that we could infer from these alignments of tens of thousands of proteins, which means that the method indeed is able to recover some kind of physical interactions, but that are basically symmetric. So there is no, for this particular data, there is no asymmetry, causal asymmetry between this variable. So that's also a good way to test the method, is to feed it with non-causal data, which you can either have in the form of this protein structure, or you can generate with the benchmarks. So we did both.
So your causal method maybe does it maybe suffer from some instability? Because if you assume a causal relationship, not just relationship, but causal links, yes. So it's a harder task, it's harder problem, yes. And so maybe maybe some instability appears or not. What is more stable? What is more stable, random forest, Genie 3, or your method? In other words.
So we've
Your
compared
method has more information.
we compare with the Genie tree. Yes, I think this is something we have to do. We do extensively, and that's actually what we do now, because we're building these very large networks. So I can't really claim right now that we are better or much better, but I think this is the case. But we'll still need to show it. To come back to your question, so we're not assuming causal relations, as I was saying, and we're just trying to discover it if there is some, and when there is none, we should, and this is something we check, obviously, we should be able to show that there is no causal relations in this data when there is none. So there is no particular instability about the assumption of causality, because as you saw, the causal discovery comes as a second step. So the first step is really to learn the skeleton of this graph without without orientations, and then when we can, we orient this graph, but it's based on information that is actually in the data. So we are fairly actually conservative here. So we only orient the edges for which we have reasonable or strong information in the data.
So okay, and the graph you found is not unique, I think. Yes, you found some of possible graphs. Yes, maybe
okay, so that's an interesting question. So the method itself gives you a unique graph because it's kind of a greedy method. But what we've done, so when you look at very large data set, the way we look at it, I mean, I have all this on slides, but I think it's better not to go back to slides. But what you can do by looking at 400,000 patients is to look at subsets of this data. So we look at 100,000 subset of this 400,000 subset set of data set. Sorry. So by looking at 100,000 subset or 10,000 subset or 1,000 subset of patients in these data sets, you will learn different networks, of course. The smaller the data set, the sparser the graph is. That's obviously expected, because the larger the data set, the more subtle effects you're able to discover. So just to give you an idea, the global graph has, I forgot the actual number, but almost 300 edges for 400,000 patients. If you go to a much smaller data sets, this number of edges will shrink. So with 1,000 patients, I think you have 40 edges or something like this. So it's really a much simpler graph. But what you can realize is that these simpler graphs, they are basically subgraphs of the full graph. So it means that by looking at subset of the data, you learn, some part of the full graph, let's say. And when you look at different instances of this 1,000 patient, indeed, you will learn slightly different subset of the full graph. So the intersection of the 1,000 patient subgraph will be significant, clearly significant, but not huge. Maybe 50% of the edges will be common in these two smaller graphs when you look at independent subset, but the two of them will be included in the full graphs to be, simple, to simplify the result, but that's the essential result that we obtain. So another way of saying it, I think, is to say that indeed there is no unique network. So when you look at a finite data set, you will learn a graph that is the good model for the data sets you provide, but if you give another equivalent data sets, you will typically learn slightly or not so slightly different graphs. But they are both good models.
Okay.
I'm not, so that's the, so in particular, in terms of causal relations, I think the causal relations will still hold when you look at these subgraphs. And we verify this to some extent, although obviously the fewer data you have, the less confident you are on your conclusion. So this is really the reason why we look at very large data sets, is to see, to push our methods to see whether, when you can't argue that there is not enough data to make good predictions, you can really test the quality of your method itself, and this is what we try to do.
Mhm. Okay, thank you.
Okay, thank you very much. So great talk, and thank you very much for answering the questions. So I think we can finish for today.
Okay. Thanks a lot, everyone.
Yeah. So video will be available on our YouTube channel, I hope today or tomorrow. So everybody can subscribe it, and I'll put the link in the chat. So and you can follow also our kind of next webinars on Twitter or on Telegram and so on. So thank you very much again, Hervé, and thank you everybody for being here and for questions. So have a
Thank you.
Bonsoir. Have a good evening.
Bye-bye.
Bye-bye.
Bye.